Hi, everyone. Today, I'm excited to share more about the science of biomanufacturing and how it can make the world a better place. Very briefly, I'm Erin, a data scientist in climate biotech. My technical background started in computational biology at Carleton College in Minnesota, after which I got my first job at Amyris, a biotech company who engineers yeast to make sustainable alternatives to petroleum-based products. I later ventured up north to UW to do a PhD in computer science, developing computational methods to decipher the genetics of methane-eating bacteria. After graduating, I was a data scientist at LanzaTech, using bacteria to convert carbon emissions into ethanol. And now I'm with SymbiBio, developing tools to better monitor and troubleshoot the complex biological dynamics in industrial bioreactors. All of these experiences fall under this umbrella of biomanufacturing, which I think is super cool, but it's also a critical lever the world can use for climate and sustainability goals. So, what does that mean? What is biomanufacturing? Let's start with the bio. There are many aspects of sustainability, but one I think about a lot is all the stuff we use in our everyday lives and how we source the material to make it. Many of the materials and ingredients we use come from biological sources. Perhaps obviously the flavorful spices in our foods, and fragrances and candles and perfumes. But let's not forget fibers in clothes like silks, medicinal molecules found in plants, and fuel, which came from a biological source that lived a very long time ago. Biologically derived molecules have the nice property that nature has saved the instructions for how to make them in DNA, which means each of these organisms has some set of genes that enable it to make a useful molecule that we have discovered and incorporated into the stuff we use. Sometimes we can harvest this molecule directly from the organism that makes it, but there's a growing field of biotech dedicated to metabolic engineering, where we can produce molecules biologically by copying genetic instructions from the original organism and installing those instructions into a microorganism. It turns out DNA is a decent API for transferring pieces of biochemistry between organisms. It's tricky, there's a lot of nuance and engineering cleverness I'm glossing over, but when done correctly, the resulting microorganisms effectively become tiny molecule factories that churn out the molecule we want. With tiny factories in hand, now let's revisit that manufacturing part. Humans have been using microorganisms as factories for millennia. For example, using yeast, which eat sugar and help us make great products like beer and wine and bread. But what if we didn't want alcohol? What if we could engineer yeast to make other useful molecules? One of the first success stories in this space was to use yeast to convert sugar into artemisinin, an ingredient in malaria medicine. This plant, known as sweet wormwood, naturally contains the DNA instructions to produce the artemisinin molecule, which does not exist in yeast. But by copying the plant genes involved in artemisinin production and installing them into the yeast genome, researchers were able to engineer the yeast metabolism to funnel resources away from alcohol and towards artemisinin. But when we're manufacturing something, we're trying to make a lot of it. Yes, the sweet wormwood plant makes artemisinin already, but you would have to farm a whole field, harvest it, and extract the trace amounts of the active ingredient from the leaves, while most of the growing energy went into building all the other leafy parts of the plant. Through metabolic engineering, scientists work very hard to optimize microbes to funnel resources towards the target molecule as efficiently as possible. With these specialized microbes, we can now use fermentation technology to brew molecular ingredients in gigantic bioreactors instead. That, in essence, is biomanufacturing. This process, metabolic engineering coupled with microbe fermentation, has been extended to produce all sorts of materials, from solvents to skin moisturizer, jet fuel to fancy fragrances, bioplastics or silk proteins, the list goes on. Nature's catalog of DNA instructions is quite vast. Okay, so biomanufacturing is pretty cool scientifically. It has infrastructure for scaling up, but how does it contribute to a better world? Well, let's review this equation we established. With sugar plus yeast plus science, we can make renewable products, which is especially exciting for replacing fuels and commodity chemicals usually sourced from fossil carbon. However, sugar carbon, though renewable, is also a crop, so it competes for land with agriculture and natural ecosystems. What if I told you you don't have to use sugar? There are other microorganisms out there. A lot of them, in fact. Microbes live in all sorts of weird nooks and crannies throughout Earth's varied ecosystems, and as such, have evolved an immense diversity of capabilities. Which means, when choosing your biomanufacturing approach, nature doesn't just have a catalog for potential ingredients to make, You can also choose from many potential organism hosts in which to make them. This diversity across microbial biochemistry creates a large optimization space for biomanufacturing strategies. Beyond the sugar-based fermentations, there exist microbes who are happy to survive on sunlight and CO2. There are bacteria that eat methane, a greenhouse gas 80 times more potent than CO2. Some microbes evolved in deep sea hydrothermal vent communities and snack on carbon monoxide. Sure, some organisms are more cooperative to work with than others, and some will have more challenging biochemical constraints. But this is all to say, the scientific search space here is quite large. There are many levers to pull as we creatively design biomanufacturing systems. I'm especially enthusiastic about biomanufacturing processes that consume carbon sources that are toxic or a greenhouse gas or otherwise a waste product from current human activities, because if we can do that well, we get to wield the double-sided saber of sustainability. Not only are we producing materials that forego pulling more fossil carbon out of the ground, but the input carbon itself is diverted out of emissions waste streams where it is instead recycled into materials we need. Both can help mitigate climate change and move us towards more circular, sustainable patterns of resource use. In addition to sustainability and climate health benefits, biomanufacturing is also quite relevant to national security. About a year ago, in April 2025, there was a report released from the National Security Commission on Emerging Biotechnology, a bipartisan group in the US government of senators, congresspeople, and scientists. The report outlines the importance of investing in biotechnology and the stark consequences to health, agriculture, and defense systems if the US falls behind in this global technology race. Biomanufacturing is a big theme throughout, especially when it comes to stabilizing domestic supply chains. If critical materials like fuels, pharmaceutical ingredients, textiles can be manufactured locally rather than imported from other nations with whom tensions may arise, not only will it provide a plethora of domestic manufacturing jobs, but perhaps you'd be more resilient to turbulent geopolitical situations. But as I mentioned, I'm a sustainability fangirl. One of my favorite tangible examples of sustainable biomanufacturing was a collaboration between REI and the biotech company LanzaTech. I'm a bit biased. I used to work for LanzaTech, but briefly, LanzaTech uses bacteria that have similar metabolisms to ancient deep sea vent bacteria, and they happily consume gaseous mixtures of carbon dioxide and carbon monoxide. Apparently, that's not too dissimilar from the composition of steel mill waste gas. So LanzaTech works with industrial facilities like steel mills to pipe their emissions into a bioreactor to feed bacteria, and the bacteria convert the carbon into ethanol. Ethanol can be used as a building block for many things, including ethylene, a precursor to polyester, which is a material used in many synthetic fiber products, especially quick-dry athletic gear, a common sighting at your friendly neighborhood REI. Now, as a Seattleite with a huge weakness for colorful outdoorsy stuff, I would already be tempted by such things. But seeing the tag mention the carbon capture details enabled by LanzaTech's biomanufacturing process was super cool. It was very inspiring for me, made for some great Christmas gifts that year, and goes to show that biomanufacturing isn't science fiction. It is possible. We can already hold its successes in our hands and hang them in our closets. In the last couple decades, a whole ecosystem of biomanufacturing companies has been growing. Many companies choose to work with E. coli or various yeast species, as these model organisms have been studied for decades and have a lot of tinkering tools already available. Some have turned to photosynthetic organisms like cyanobacteria and spirulina. Others go that gas fermentation route, like bacteria that consume methane or carbon monoxide. And some are even branching into algae and mycelium-based hosts, or intentionally working across the spectrum to stay organism agnostic. Great, sustainable biomanufacturing is here. We're done, right? Sadly, no. The work is not done. If you were to take a tentative internet stroll amongst these names, you may come across some disheartening tales of bankruptcy filings, layoffs, hiring freezes, stock price trends that are sadly reminiscent of certain playground equipment. Many promising biotech companies have come and gone, often stumbling in that proverbial valley of death between early proof of concept and eventual commercialization. This is where many struggle financially to make ends meet. Companies have progressed beyond that prototyping phase that is often supported by startup investment, but they are still working out the kinks of their processes, and they are not yet able to produce their target molecule efficiently enough to be profitable on their own. A reason this valley of death is so perilous in biomanufacturing is because the scale-up process itself introduces new scientific variables that were not encountered during prototyping. As a couple concrete examples, when you're testing molecule production in the lab, you may see couple liter tanks happily spinning its microbial broth with small propellers to mix around the food and nutrients. But when you're running tanks that are the size of 10-story buildings, it would take far too much energy to spin a propeller to mix hundreds of thousands of liters of broth. So the distribution of food and nutrients is a lot more heterogeneous within the huge reactor, limiting the overall availability of energy resources to the working microbes. Similarly, if you're trying to clean your bioreactor between runs, you can stick a half-liter fermenter in an autoclave, a fancy science dishwasher that sterilizes lab equipment. There is not an autoclave the size of a building, so cleaning procedures are more akin to brushing your teeth between runs. It helps, but it's not perfectly sterile, and so large-scale processes have to be robust enough to operate within more varied microbial environments. These kinds of variables can throw off the correlation between how efficiently processes work under small-scale conditions versus large-scale conditions. Ideas that looked promising in the lab can turn out to be duds when put in the industrial environment. Many biotech orgs have learned the hard way that promising overly optimistic production numbers based on lab-scale data can leave people a bit disappointed and, frankly, more wary of trusting biomanufacturing ventures as viable, profitable businesses. Now, my point in sharing this is not to rain on the biomanufacturing parade. This is hard stuff. Biological systems inherently have some degree of evolutionary agency to change in ways that pure software and circuit boards do not. My point in sharing these challenges is to highlight that there's huge opportunities for improvement. I recently learned of Wright's law, a formalization of a learning curve, which describes the relationship between expertise gained over the cumulative production experience of making things versus the steady overall decline in production costs. Wright developed this in 1936 while studying manufacturing of airplanes. You may have heard of Moore's law, which is similar, but it predicts decline in production costs as a function of time, whereas Wright's law is a function of experience. Basically, we learn by doing and gradually figure out how to do it better and cheaper. To descend this learning curve, biomanufacturing companies need the opportunity to gain that cumulative experience at scale. After all, the first biomanufacturing companies to target specialty molecules beyond just a single enzyme or protein only got industrial scale operations off the ground in the last 10 to 15 years. We've been manufacturing things like planes for over 100. There is a lot of learning from experience still to do. But most especially, I think there are huge opportunities in this space for better learning from data. All right, data people, this is your biomanufacturing bat signal. Data people are essential for tasks across the life cycle of biomanufacturing processes. At the very beginning of metabolic engineering projects, companies need to decide if a molecule is even worth manufacturing. They need to know what's the biochemical feasibility of making this molecule with this organism host using this feedstock. This is known as constraint-based modeling and is usually helpful for deciding which projects to pursue. Since it's way too expensive to run suites of experiments to test ideas in 10-story buildings, you have to try a lot of stuff at small scale first. Thus, we need automation engineers that can interface with the robotic equipment that enables people to test thousands upon thousands of combinations in high throughput. After screening thousands of combinations, we next need people trained in statistics to help analyze said screening data and find the hits, such as organisms or conditions or process parameters that produce significantly higher yields of your desired product, and then iterate on the best ones for the next round of trials. As I mentioned, correlation between small-scale performance and large-scale success remains tricky. It's sort of an Achilles heel for many in biomanufacturing. So getting talented brains in here to figure it out is crucial. Did someone say iterative learning from many examples? Smells like machine learning to me. Each of the companies I showed probably has tons of data. Some are arriving at a couple decades that they have accumulated. Developing more sophisticated predictive models from time series data of production runs could really help companies monitor and manage their live biomanufacturing processes, which are very expensive to restart if things go sideways during fermentation. Relatedly, there's a lot of questions around the status of the microbial strainer community used during a fermentation. Which microorganisms are actually present and active, and is it the one you expected? How is the population changing? Are they mutating? How is all of that affected by other process variables? And of course, underpinning all of this is data engineering. From all sorts of measurement equipment, we need people establishing proper data collection and processing pipelines, storing it efficiently in databases, and developing intuitive access paths so it can be found and analyzed. Data engineering can sometimes be left by the wayside when there's pressure to move fast on the biomanufacturing technology itself, but it really comes back to bite. When you want to compare current operations to data from 5 years ago and it's buried somewhere in SharePoint rather than a database, when you want to assess the efficacy of a certain nutrient supplement, but it's only been recorded as a free text comment, often without units, when you want to use historical data for machine learning, but periods of performance were never formally labeled as good or bad outside of the moment they were happening. And so sometimes data are not yet usable for training models to predict performance. I firmly believe that more data people need to be in the room for biomanufacturing processes to reach their full potential. As they are designed, as they are executed, as they are reflected upon in postmortems, extensive data collection, infrastructure, and integrity are critical to enable the learning from experience that biomanufacturing needs to become a more reliable and resilient field. As a final note for this biomanufacturing story, I want to highlight a critical data stream that for now is typically missing, and that's real-time biological sequencing data. Most biomanufacturing systems will have pretty much real-time data for system inputs and outputs, such as the flow of carbon and nutrients in, the amount and rate of target molecules being produced, and other process data like temperature and pH. These data streams are closely monitored by biomanufacturing operators, the people in control of all the dials, pumps, and levers needed to run a 10-story building-sized bioreactor. It's their job to keep the microbes happy and growing and producing your target molecule. Occasionally, these production numbers start to veer off course, and operators do their best to quickly troubleshoot and adjust the necessary dials and pumps and levers to restore the system to stable production. When they can't restore stability, fermentations have to be shut down and restarted, a very expensive and frustrating process. But operators are generally blind to the actual biology inside the reactor, the process underlying the molecule production. Are the microorganisms you expect actually present, or is there a bad guy population starting to take over? This could be hugely valuable information as operators make troubleshooting decisions. Biological data is possible to collect. You sample the broth, send it out for sequencing, and get some computational biologists to take a look, but it's typically not part of operators' active decision making. Even when sequencing data are collected, it's usually not available until several weeks later, after which the bioreactor has moved on and is in a totally different state. This means that those sequencing data can only really be used for post hoc root cause analysis to understand why operational failures happened, but can't be used to actively prevent failures before they happen. To fill this critical gap in real-time biological information for biomanufacturing, that's what I'm working on right now with SymbiBio. We're fairly new to the party after a tough Valley of Death / DOE grant cancellation combo that triggered layoffs at LanzaTech last year. We're a startup rising from some of those ashes. A clear window into the biology could empower operators to make more informed decisions about how to keep their biomanufacturing processes running smoothly and avoid costly losses. We're currently a team of 4 scientists and engineers who dealt with many of these scale-up challenges firsthand and are now building out software and genomic measurement systems to cheaply measure, continuously monitor, and eventually predict microbial dynamics in industrial biomanufacturing settings. We are very excited to help companies trudging through that scale-up valley of death actually come out the other side. And with that, I'll conclude. I hope you've enjoyed some background about biomanufacturing. It has a lot of promise for creating renewable domestic supply chains for critical materials and ingredients that we use every day. And furthermore, this industry could really use the skills and experience of data experts to help optimize biomanufacturing technology. That way, we can continue shifting humans towards a more sustainable relationship with the resources on our planet. Thanks for listening, and I hope to see you around the field soon.